This is the final video in a four-part series on how to perform correlated light and electron microscopy with mini SOG. This video will cover how to prepare your photooxidized samples for electron microscopy with a brief guide to interpreting results. At this point, you should now have photooxidized samples and consolidate buffer. The first step is to fix your sample with osmium tetroxide. Be careful. Osmium tetroxide is very dangerous and must be used in a properly ventilated fume hood. Stock 4% osmium tetroxide must be diluted down to 1% in 0.3 molar consolidate buffer. The recipe used is seen here. Carefully replace the consolidate buffer that your sample is currently sitting in and replace with the 1% osmium fixative solution. Place your sample on ice and let fix for 30 minutes. After osmium fixation, we typically stain our samples with uranyl acetate. This gives additional contrast as it labels proteins generally. If the signal from the photooxidation is weak, this step may be left out so that more contrast comes from the photooxidized area. Before using the uranyl acetate, we always freshly filter a solution of 2% UA before staining. After the 30-minute osmium fixation time has elapsed, remove the osmium and wash 3 times with water. Replace the water with your filtered uranyl acetate solution. UA staining takes at least 1 hour, but can be taken overnight, as long as the sample stays at 4 degrees Celsius. We usually use 3-minute washes of 20, 50, 70, 90, 100, and 100% cold ethanol in that order. This process is finished by one final wash with 100% pure ethanol at room temperature. After dehydration, the sample is embedded in resin. Here we use Durkapan epoxy resin. Proper mixing is key. As a rule of thumb, we stir 100 times in each direction. Once completed, make a mixture of 50-50 Durkapan to ethanol and mix thoroughly. Remove the 100% ethanol from your sample and then place your 50-50 mixture on top. Incubate on a rocker for 20 to 30 minutes. Afterwards, replace with 100% Durkapan. Scrape out and replace the Durkapan 3 more times, at least 1 hour each, making 4 resin changes in total. Finally, bake your sample in a 60° centigrade oven for at least 48 hours. Your sample is now ready for sectioning, and then imaging with your transmission electron microscope. Here we will show some examples of successful minisog photooxidation. Histone 2b, one of the main histone proteins involved in chromatin structure, was tagged with minisog. As expected, minisog fluorescent signal was observed in the nucleus. Note the areas of high signal density became the darkest after the photooxidation reaction. Here, at high magnification, where normally the nucleoli would be the most electron dense, we can see this very fine granular stained material representing the H2B in the nucleus. Here is another example of the application of minisog. Here, gap junction protein connexin 43 was tagged with minisog. On the left-hand side, we can see the fluorescent signal localized between cells, and after the photooxidation reaction, these areas are specifically stained. When brought to the electron microscope, density is observed where it is expected, between the cell bodies. At high magnification, the ultrastructure of the gap junction is visible. This concludes our 4-part video protocol on how to perform correlated light and electron microscopy using mini SOG. These videos were filmed and produced at the National Center for Microscopy and Imaging Research at the University of California, San Diego for the UCSD Interfaces Graduate Training Program, with support from the Howard Hughes Medical Institute and the National Institutes of Health.